Um, so before I jump in to this project, I'm mainly going to be talking about the diatoms, what diatoms can tell us about sea ice in the North Pacific Ocean broadly. And I just want to thank all of my collaborators, in particular, the three at the top, Kate, Anna, who is on this call, and Summer Pretorius. The four of us are working on a publication about this, and so that should hopefully be coming out at some point. And I also want to recognize my PhD advisor, Julie Brigham-Grady. She advised my master's and my PhD, and I don't know if any of you know her, but if you do, it won't surprise you to learn that Julie thought this would be a good master's project for me, and it's like 20 years later and I'm still working on it. So it's a very Julie thing. There's a lot of other people who've been involved with sharing data and working on, talking about proxies and things like that. The students at the bottom were integral in helping do some of the counts for some of the diatom records that are in this. And then I just want to also recognize my funding comes from the USGS in the Climate R&D program, and also an NSF award that's hosted at University of California, Santa Cruz. So to jump right in, the motivation for thinking about sea ice and how sea ice has changed in the past comes from what's happening today. And when we look at sea ice, this is an oldie but a goodie. I love this animation. It started in 1979, and it's showing you the aerial extent of sea ice in the summer. So this is the minimum amount that sea ice shrinks back every year. And, we see this precipitous decline, particularly once we get into the 2000s, it starts to really go down. And so, this is part of the climate crisis that we're all experiencing right now. And it makes me think, has this happened before? My gut wants to say no, but as a geologist, I want to look back in time and sort of investigate that. Have we had a precipitous decline in sea ice in the past? Has the Arctic Ocean been ice-free in the past? What if we go to a time period when the Earth warmed naturally, like from the glacial into an interglacial? What does that look like as sea ice disappears? There must have been more sea ice during the glacial than there is today. So how did that change? And how does sea ice interact with primary productivity? And so it's sort of a chicken and an egg with the primary productivity, like how does sea ice impact primary productivity? But we're actually going to use primary producers in order to figure out what sea ice was like in the past. And so my goal is really to sort of extend this record, extend it back thousands, tens of thousands of years so that we can have an idea of what, so we can really put this in context. So the project that I'm going to describe to you, this map showing across the Pacific. Can you guys see my mouse if I do this?
Yeah.
Okay.
Looks great.
So this is the Bering Sea, this is the Chukchi Sea, and then of course the open Pacific with Alaska and Russia. And so we have 59 records from 52 sites across the Pacific and the marginal seas, the Gulf of Alaska, Sea of Okhotsk. We've got some sites. This one is right off the coast of Tokyo, and these two are right at the California-Oregon border. So I'm kind of excited about those three sites. So all of this is almost completely a literature compilation. So the data is stored usually in publicly available repositories. Some of it is things that I've had to request from people, and there are a few records of my own in there as well. We've been working to update all of the age models to the most recent Marine 20 calibration curve so that the dates are all so that we're, looking at dates that are the same. We're not comparing different calibrations to each other. And then the idea is we have 12 time intervals, and we're taking the mean sea ice coverage for each time interval at each site. And so this is a little bit problematic, and this is what I'm going to get into, because sometimes if you have a site that has more than one proxy or more than one type of record, they don't agree. And so we have to figure out, okay, what are we going to choose here? And our strategy has been to look at what the original interpretation of the data was by the original author, as well as look at what the different the limitations of the different proxies, and choose the one that seems like it's the most appropriate for that site at that time period. And then the final product is to make some maps, and so I'll show you those at the end. So if we look at the literature, we look at what people do in order to reconstruct sea ice. There's 6 main types of proxies that people use. And this is the distribution of how they break down in our study. I'm going to focus on these 3 really in detail in this talk. I'm going to get into each one because these are the 3 that relate to diatoms. And so this is what diatoms can tell us about sea ice. There are 3 non-diatom ones. So I'm just going to tell you quickly about those. There are some studies that use dinoflagellate cyst assemblages, and this little guy is Landinium minutum. This is a dinoflagellate cyst that is associated with sea ice. There's a new proxy. It's an alkylone-based sea ice proxy, and the basic idea here is this is a molecule that is produced by a primary producer that lives in sea ice. But this primary producer is not found in sediments. It's sort of a relative of a coccolithophore. And this is a very preliminary proxy. There's only been about 2 papers published about it so far. But if you look at the abundance of the molecule in relation to sea ice, it looks like there is a relationship. And so I was excited to bring this in and just see how it compares to the other proxies. And then ice rafted debris, which is, as sea ice is forming, it can pick up little stones from the seafloor and entrain them in the ice. The ice will sort of get blown around, and those little rocks stay stuck in the ice. When the ice melts, the ice rafted debris melts out, and they're left for us to see. There are a lot of IRD records, and a lot of them are problematic because at a place where we actually are sort of on the margin of the continent, it turns out there's a lot of ways that you can get coarse debris in these marine cores. And so I tend to not prefer that one if I have other options for proxies. Okay. So we're gonna dive right into diatoms, and the first thing, I don't know, I feel like a lot of you are modern people, and so some of this might not be familiar to all of you, but if you're a paleo diatom person, this is a super familiar thing. So this is a standard kind of plot where you have time on the y-axis, and then we stack up a whole bunch of different diatom species right next to each other and look at their relative percent abundances and how they change. In this case, I've colored these blue ones are all sea ice diatoms. And so we can really get into this and say, like, how did sea ice change over the course of this record? And that's great. And, it's definitely something that we're looking at in this project, but with, almost 60 records, we want to try to simplify this a little bit. And is there some way that we can take this qualitative assemblage and sort of apply it to a lot of studies? And so what we're doing is we're focusing on the genus Fragilariopsis, which is these first 2 right here. And looking at just the percent Fragilariopsis. So Fragilariopsis has been associated with sea ice and used as a sea ice indicator for many decades, maybe even a century now. So it's pretty common in counts, old and new, which is great. However, there are some problems with it, of course. Taxonomy has changed a lot over time, and now we know there's actually 11 species of Fragilariopsis in the Arctic. Do they all relate to sea ice? No, of course not. So that's a problem, especially since a lot of people just count Fragilariopsis SPP and put them all together. Anyway, so we looked at that. This map here is showing, I was going to switch these around. Anyway, sorry. This map is showing the relative percent abundances of Fragilariopsis in the Bering and Chukchi Sea, and you can see the redder colors My cat has arrived. I don't know if you can see her, but I'm gonna work hard to not let her sit on my computer. The red is where we have the greatest abundances of Fragilariopsis. And if you can imagine, I had a map of sea ice concentration that I'm going to show you in a little bit. And what's up over there? If you could see that, you would see that sea ice starts here and it increases in concentration as we go north. And so this area here is sort of a sweet spot of about maybe 50–60% sea ice in the springtime. So on the other hand, there is a lot of Fragilariopsis in places where we don't have sea ice, so it's not perfect. If you look at the percent abundances of Fragilariopsis in the sediments, and these are just box and whisker plots for different bins of number of months of sea ice present per year. Again, it peaks right around this 5 or 6 months of sea ice per year. And so these are just a couple examples of what Fragilariopsis looks like. And this one here on the right, Fragilariopsis reginianae, this one was described in 2000. And so it wasn't included in all of the very old counts. So that's one of the problematic ones. Your guess is as good as mine, what it was counted as, because it's pretty common. We also have this one. Oh my goodness. Can you guys hear the drilling in my wall, or are
Not
my
at all.
headphones? Oh, really? Okay. There was just like extremely loud drilling and I couldn't hear anything. So fabulous. I will just continue on and pretend that I can't hear it either. So this species is Fossiloficus. It's not even a Fragilariopsis, but it was described in 1992. And so it also has, I think, been dumped into these Fragilariopsis counts. The good thing is this one actually does really like sea ice, and it can be up to 70% in some of our assemblages. And so that's great. So anyway, okay, qualitative diatoms. We're going to move into quantitative diatoms. This is work that Anna Nestrovich did during her dissertation. And sort of the idea was to take this full assemblage and try to make it absolutely quantitative. And so here's that plot of sea ice concentration from no sea ice. And we get the sea ice margin here, and we increase up to 100% sea ice coverage in the springtime. And so she looked at the assemblages and all of those dots, and then she used the random forest method to pick 10 species that have a relationship with sea ice. And then we looked at those 10 species and we're like, okay, what do we know about the ecology of them, and which ones are maybe, people don't really count them, or they're pretty rare, like which ones can we get rid of? We settled on 5. And there are 6 here because the model either uses Fragilariopsis virginianae or Fragilariopsis oceanica, depending on whether it's a new or an old count. And so these diatoms, this is Cinerema marigella, and it has the most affinity with sea ice, and the affinity sort of decreases as we go down here to Neodenticulus seminae, which lives in open water. And so with those 5 species, she then used a generalized additive model to take those percentages and turn them into spring sea ice concentrations, which is a great thing because now we have a number rather than just saying, oh, there was sea ice present, there was no sea ice. We can say there was 50% sea ice in the spring, or there was 80% sea ice in the spring. And it comes with 90% confidence intervals. So it gives us an idea of how well the model is performing as well. So just to show you some results from this, this is this green site up here. This is a site that both Anna and I counted, and it uses her full model with both Virginianae and Cinerema, which are really new to the taxonomic literature. So what we realized is that there's a lot of old counts out there from diatoms going back, into the 80s, that it would be great if we could include these because they have a lot of really fabulous information, and it's not really practical to recount them. And so Anna played around with trying different models, looking at what were the diatoms that they included in their counts. And okay, if they didn't include Virginianae and they didn't include Cinerema, like what can we use instead? And she came up with 10 different models. Some of them work really well. Some of them not as well as others, but we were able to sort of tweak that model a little bit. This is this site right here. And it's a little bumpy, but I think that is probably reasonable. This is Bowers Ridge, which is out here. This is a site that probably never has sea ice. And the model is showing us that it has pretty low concentrations in sea ice, but it also, this blue is the error bars. So it's like, model, I don't know whether model number 6 is not working that well, or if Bowers Ridge is just not working that well, or if these counts, I think remembering back to this study, there were not a lot of different taxa included in the counts. So that was a bit of a challenge. So I'm just putting this out here to say, like, there's some challenges. It's not 100% perfect, but there is a lot of information we can get. And then the last thing about this proxy, which we're really excited about, but we want to just sort of underscore that it probably doesn't work outside of the Bering and Chukchi Seas, because that's where the proxy was trained. And, there may be other species that are dominant in assemblages in other places. So the third diatom proxy, this is sort of an indirect diatom proxy, IP25. This is a molecule that is biosynthesized by diatoms. It's a highly branched isoprenoid. IP25 stands for ice proxy with 25 carbons. So this molecule up here has 25 carbons. It's been found to be biosynthesized by things like Haslea and Pleurosigma. And they're not ice obligate, but they are commonly found in the ice. And when they're in the ice, they create IP25. But the other thing about them is that they're these giant, easily broken, really fragile, finely silicified species. And so we don't usually find them in the sediments. Often I'll find like just a little square of, like the areola in here and be like, oh, I have a little bit of Haslea, but it's not enough to actually count. And so that's interesting. This is like a diatom proxy, but it's completely different from the assemblages because these never show up in the assemblages. So one of the drawbacks of IP25 is that it's present under seasonal sea ice, which means if you have open water, you have no IP25. And if you have perennial sea ice, you have no IP25. And so this is a problem if you think you might actually have perennial sea ice at some point. And so in order to address this, what people have done is they've coupled the IP25 with a biomarker from open water phytoplankton. And so in this case, we have IP25 on the y-axis and brassicasterol on the x. And each one of these is a sample for different places. And basically you can take the ratio of the two and normalize it. And then the PIP 25 is what it's called, is what we'll plot on these curved lines here from that equation. And it basically divides the sea ice into 4 different types of sea ice, ranging from extended to marginal, less, and then ice-free, based on what the value of that PIP is. And so this is another thing about this project is we've got some proxies that tell us presence and absence. We've got some proxies that tell us a concentration of sea ice, like what percentage of that area of water was covered in sea ice. And then we've got some that say, oh, it was marginal ice or it was less ice. And so there's these proxies are sort of reconstructing different things, which sort of adds in an extra challenge. So here come some results. I'm going to show you a few sites that have multiple proxies in them and show you how they compare. And so the first one is that star here in the Bering Sea. And the purple is the GAM, the quantitative diatom assemblage. The blue is percent fragilariopsis. Those are both on this axis. And then the green is the PIP 25 on this axis. And what you see is both the GAM and Fragilaryopsis show us that there is sea ice until about 10, 11,000 years, and then it decreases and goes away. The Fragilaryopsis is low out here, and so even the GAM is showing high sea ice. There are different sea ice diatoms that are prevalent at this point in time. So Fragilaryopsis is low. But the PIP actually says, nope, sea ice disappeared around 16,000 years ago. So this is a huge problem, and this is really common in a lot of the sites where we have IP25 and we have diatom proxies. They don't agree very well. There's an offset in the timing of when they show that ice is decreasing. On the other hand, these two sites are from the Gulf of Alaska. So the triangle at U1419 in this square, this is actually in a fjord. This is at the mouth of the Bering ice stream. And so there was like a ton of glacial outwash in there during the last glacial. And what we see here, again, the purple is the GAM, or the quantitative diatom. The blue is percent Fragilaryopsis, and the green now is alkanones. And in this fjord site, they all agree. This is lovely. It looks really great. Out at 1419, it's a whole different story. The GAM is out here. I don't know what the GAM is doing. It's got like from 0 to 100% error bars. There's one peak in Fragilaryopsis really early on, and then nothing else. But the IP25 shows extensive sea ice, and then it disappears. And the alkanones also show sea ice, and then it disappears. So in this case, I think what's happening is that this massive glacier is just like pouring a ton of meltwater, as well as ice rafted debris and all kinds of things out into the water, and really creating sort of a different environment for diatoms. And the diatoms are really, really sparse in this core too. So I don't think the diatoms are working all that well. It is interesting to me that the IP25 and the alkanones seem to work okay. But this also raises a point, which is these massive error bars. I think they're just also showing here, you know, the Gulf of Alaska is not really the appropriate place to be using this quantitative diatom proxy. And so we would get rid of that too. So there are some differences. There's differences in between, you know, the IP25, which is based on diatoms, and the diatom assemblages. And so what could account for these differences? And I put this picture up here. It's not a perfect analogy, but I think it's okay. This is a picture that was taken last spring off the coast of Nome in Alaska. And what I love about it is that it shows how different sea ice looks. I feel like most of us have very little experience with sea ice. And we probably imagine something like, you know, the massive ice off of Antarctica, or these big walls of ice, and you have this ice margin. And that's not what sea ice is like in the Arctic at all. So sea ice is pretty thin. It's like a meter or two thick, and it forms at the coast, and then it gets blown out and moved around. And, you know, you can see all of these are called pancakes, all of these round things. And so the pancakes will move around, and they'll pile up against each other and form bigger pancakes. And so it's a really dynamic environment. My cat is back, and she's really used to sitting on my computer. And what we see here is right along the coast, we have complete, it's probably fast ice. So it's solid ice. It's attached to the seafloor. And then as we move out, we have thick ice here, but you can see it's made up of a lot of different pancakes. And so this has probably been blown in towards shore and piled up, and sort of gotten thicker and thicker as it piles up. And then as you get out here, there's like a mix of sort of thin ice. There's some open water in here. There's leads between the ice. And so there's probably some melting going on. And so if you think about that, you can think of these as 3 different habitats for different types of diatoms. And maybe the IP25 producers are going to be living attached. I mean, they have a reef. They like to attach to the underside of ice. So maybe they're living, you know, out here in the fast ice. Whereas there are different species that are living out with the pancake ice, and then Fragilaryopsis is out here in the melting ice. So we do have a little data that supports this. This is also Anna Nestorovich's data. And so just to sort of walk you through this a little bit, the yellow is sunlight. And so we're starting, this is 1 year of data from sediment trap in the Caissie Sea. And we start in August and we go until the following August. So sunlight is declining. We get to polar night here in November for a couple of months. Sunlight comes back, and then we have 24 hours of daylight before it starts to decline again. And then on top of that, the gray is the sea ice. And so we have no sea ice in the beginning for several months. Sea ice freezes up pretty quick. We have pretty solid sea ice for 6 or 7 months, and then it decreases a little bit more slowly than it froze up. And so we can look at the diatoms, we can plot them on top of here. And this red one is, and it's clearly, Nichia longissima, it's clearly an open water preference. And so this is one that has a really clear preference. A lot of the other ones didn't have a super clear preference. However, there were some changes a little bit. And so the ones I've put in here, the green ones are 2 different Fragilariopsises, and the blue ones are Haslea and Pleurosigma. These are the IP25 producers. And if we just look at when are these peaks, I mean, these are really low abundances. It would be great to have a few more years of data to find out, like, do we always have this low of abundance of these IP25 producers, or was this a particularly low year? We don't know the answer to that. And so, but what we do see is we see some peaks here when sea ice is extensive, but there is sunlight. So there's sun getting through to let them photosynthesize. So that's where I would have predicted we would see an increase in them. With the Fragilariopsis, it's a little more complicated. Fragilariopsis virginianae shows a clear peak as the sea ice is decreasing and there is a lot of sunlight. And so this is showing a preference for the melting sea ice. Fragilariopsis cylindris is peaking a little bit earlier, so that's a little bit of a surprise to me. I meant to plot it on here and I forgot, but we also have Fossula arctica. It actually, so this scale is only up to 12%. Fossula arctica gets up to like 70%, and it's going to peak. Anna can correct me if I get it wrong, but it's going to peak somewhere in this range too, where sea ice is decreasing. And so there's a lot of meltwater. And so this, I think, starts to get into what could cause some of these differences between IP25 and the diatom assemblages, is that we're looking at diatoms that have preferences for different types of sea ice. And if you can imagine, extensive sea ice with only maybe a couple months of breakup each year, you might have more opportunities for the IP25 producers to be attached under the ice, whereas you might have a shorter time span where the diatoms that like to live in the water around the ice as it's melting would have that habitat to live in. And so I think that's something that hasn't really been explored at all, and is a kind of exciting thing to do as a next step in this process. Oh, right. And then I had these images to just sort of drive that home. So this is an image of an icebreaker has gone through, turned over the ice. These are diatoms on the underside of the ice. I haven't melted this myself, but I predict if you did, it would have a lot of Haslea in it. I might be wrong. However, this is ice that I have melted myself. So a couple months later, the ice starts to melt a little bit more. There's a lot of meltwater around it. This is an image. It looks just like a dirty piece of ice, but there's a lot of diatoms living in this. And so I melted this, and this is images from that ice melt, and it's just full of Fragilariopsis, as well as Fossula arctica. So that's exciting. Okay, so just to sort of wrap up, and we can take these proxies and then apply them to all these different sites, all 52 sites. And so the first question that I asked is, when did sea ice disappear from the Pacific? So if you look at this plot here, each one of these spots is coded by what kind of proxy I'm using to draw this conclusion. And then the number is thousands of years ago. And so basically the pattern that we see is that, you know, if there's sea ice down here, it disappeared 16,000 years ago, as opposed to the Western Pacific. There's actually ice off the coast of Tokyo until 12.8 thousand years ago. And there's also this east-west difference in the Gulf of Alaska versus out here sort of in the Western Bering Sea, where sea ice tends to disappear in the eastern Gulf of Alaska around 15,000 years ago, maybe 14,000 years ago, just before the period called the Bowling-Alarod, which is a big warming. Whereas it actually comes back over here and sticks around a few thousand years longer. It sticks around on the coast, the slope break in the Bering Sea until sort of the middle Holocene. It's still present in the Chukchi Sea today, and it's still present in a lot of the Sea of Okhotsk, but there are some places where it disappears sort of in the mid Holocene a few thousand years ago. And then this map is kind of weird, so I'm going to let it run a bunch of times. But what I've done here is I've combined sea level with sea ice. And so that's the last glacial. Sea level is dropped. That's the Bering Land Bridge right there, right here. And so as sea ice rises, as sea level rises, sea ice changes a lot. And so as you sort of watch this, there's probably too much to sort of describe about what all the colors mean and everything, but the clear dots are no sea ice and the colored dots have some amount of sea ice, and that concentration differs. And so what we basically see here is that we start out in the glacial with extensive sea ice down both coasts, ringing the Pacific. It disappears in the Bowling, and then it comes back briefly, especially in the west, and sticks around, and then sort of becomes close to modern at about 4,000 years ago or so. This site down here is a little bit weird and controversial. So there's two sites. There's one that's farther offshore that has an alkanone record that shows that there was sea ice. And then the one that's closer to shore has a diatom record and has absolutely zero sea ice diatoms in it. And so I'm really fascinated by that site. I don't know what it means, but I'm also sort of captivated by the idea that we could have had some evidence for sea ice as far south as the California border. A lot of people think that is impossible. So I'd be interested to hear what other people think. And then I'm just going to leave this map up here. This just is basically that GIF. It's each panel from the GIF. So if you want to take a look at it, you're welcome to, and look at the differences between each time period. It starts at the last glacial and then goes forward in time until today. So thanks so much.
Thank you, Beth. That was really, really interesting. And don't worry, there was not any significant cat or hammering interruption. So it's
oh, that's amazing. My cat was really like, I'm gonna sit right there, and then yeah, very loud drilling.
It was good. So I guess I have some questions right off the bat. And before I ask you about reconstructing in freshwater, I'm just taking some of the things that I'm observing from our very low snow winter this year. And I'm wondering about how the clarity of the ice and the amount of snowfall, would influence this sort of thing. And again, I understand we're not looking at like one season, it's multiple, hundreds of years, perhaps represented in a segment. But, do you know from any of the modern analogs whether the diatoms are responding to this, or is light penetration good enough that they don't care?
No, they totally care about snow. And so I am not 100% up on the modern primary productivity under sea ice studies, but what I can tell you is that snow depth is very important, and the thickness of the ice is also important. And so it's not a huge problem in an area like the Bering Sea because the ice never gets really thick, but occasionally they'll crash into each other and create these big pressure ridges, and so then those are too thick for sunlight to get through. But the way I think about it also is that it's patchy, and so like you said, it's a few hundred years of time. And so, you know, if you've got 1 year with a lot of snow, it probably doesn't matter. But if it always is snowy there, then you're probably going to have really decreased productivity.
Sure. Mark, you have a question?
Yeah, Beth, cool talk. It's neat to see diatoms in a place that I don't think about them very often. Just a couple questions. That data set that you had with the sediment traps, is the sea ice extent is that all monitored via satellite?
For that site, that sediment trap has a bunch of different monitoring devices. And so we actually had the sea ice presence or absence was monitored at that particular site. It wasn't a broader regional monitoring.
There's a sensor that monitors the sea ice I assume it's underwater, right?
It's underwater and it has a thickness. Anna, do you remember what that sea ice monitor was? I'm going to put her on the spot.
Uh-oh.
Oh.
I don't think I do.
Oh.
Yeah.
All
should
right.
look that. I'll look that up and let you know.
Okay.
But I think they measured light. Measured light from the underneath the ice.
Is that what it is? It just
Well, they
measures
did measure light, but that was a different sensor. They measured light. I know they measured the thickness of the ice, and so like I know that the ice was like being measured in situ, but yeah, well, I have to double check what that is.
The other question, some of your final figures that showed these icebreakers and these big chunks of ice. Is there do the diatoms eventually actually lend themselves to the destruction of the sea ice because they're so dark and being there? Sort of like the issues they're having with like the deglaciations in Greenland, where the dust that's being created is basically melting causing even faster melt of stuff. Do you think that is also happening with the diatoms in the sea ice situation, where that's actually helping the collapse, hurting the collapse, whatever?
Well, so I think there's two different things in there. So yes, on a seasonal basis, they absolutely help the sea ice to melt. On a like global sea ice decline basis, I don't think the diatoms are having very much impact, because you do have to remember that sea ice will expand and contract every year. And so every year as that ice melts and the diatoms, start to bloom in the springtime, like you can see ice flows that have pits in them at the same spots as they have like clumps of algae underneath. And so they definitely help with that sort of short-term melting, but I don't think that they're necessarily part of the broad. I've never heard anybody say they're part of the broader sea ice decline.
Okay. Okay. Can I ask one more question?
Yeah.
Okay.
I mean, it's okay with me.
You're focused. You were focused kind of exclusively on the Bering Strait and the Pacific Ocean there. Would you be able to play with these same diatoms over on the Atlantic side of things? Is there that any sort of that level of global biogeography with these sea ice diatoms?
There totally is. Yeah. So actually, I'm part of a group that is looking at Arctic diatoms, and I was actually surprised at how similar the assemblages are in the Atlantic as they are in the Pacific. It was kind of astonishing to me to look at their counts and be like, oh, maybe our proxy would work in other places. I think the big difference between the Pacific and the Atlantic is the way the sea ice works. And so in the Atlantic, you have like export of sea ice from the Arctic. It's being shot out of Fram Strait and pushed into the Atlantic, and then it gets mixed up with all the icebergs from the Greenland ice sheet. And so you just have a very different situation. Whereas in the Pacific, there were never really these massive glaciers, except at the end of the last glacial, when you had the Cordilleran ice sheet right there. But even then, it's hard for me to imagine that. I mean, were there like flotillas of icebergs like there are in the Atlantic? I don't know. Maybe there were during like the Siku events when there was a lot of meltwater, but Yeah, so the assemblages are similar. And, you know, IP25 was actually developed in the Atlantic, and then they were like, oh, this works everywhere. So there's a lot of similarities.
Is there, with that IP25, do they know enough about it to, I mean, is it produced by every species of diatom, or, and
No.
is it, it's not?
It's just Haslea and Pleurosigma, which is the problem.
Wow.
And though they produce it when they're in sea ice. So you can have those diatoms not in sea ice and they don't produce IP25. So it's like, they don't know what the mechanism of that molecule is, but it's got to be something that helps them attach to the ice or helps them not freeze, or like it has something to do with them being in the ice.
It sounds like something to help them not freeze, at least from the name of it. Sounds a lot like what I put in my car's radiator.
Exactly.
All right, thank you.
Thanks, Mark. Sarah, you have a question.
Yeah, thank you, Beth, for a really great talk. This is really interesting. And one of the things I picked up on is you said it wasn't practical to recount past slides. Are they available? What's the archive story about what's been done from past cores?
Some of them are available. Some of them are not available. So that it's a mix, and it depends on who. I mean, some of them, even getting the data is complicated. There have been some where I'm like, can I get your diatom data? And the answer comes back, which species? I'm like, I want them all. You know, don't just cherry pick a few species for me. Give me all the data. So yeah, I mean, it is definitely a mix. Some of them could be recounted. I have some of the slides for some of these sites, but a lot of them, I don't know. I haven't done the research to see if anybody's archived them anywhere.
If the slides,
Japanese ones, or, you know, I'm not sure.
Yeah, so important to have archives.
Yes.
Great. Thank you. Yeah, I appreciate hearing about this.
Beth, I have maybe more of a nerdy technical question. But how long were the cores, and what did you use to collect them?
Oh, so this is mostly cores that I didn't collect.
Okay.
Yeah, so it's literature, it's, you know, cores that have been published in the literature. So yeah, there are a variety of lengths, but I don't know exactly.
I just think about the variability in trying to get, you know, 200 years of lake mud, and it's like, oh man, how long of a core would it be to get these reconstructions? So
yeah, and some of them are, I mean, some of these cores are like 1.5 meters long, and you know, some of these cores are many meters long. You know, I mean, there are some that are like really deep cores from drilling, and so those are hundreds of meters long. But we're probably only looking at, you know, a few meters within it.
Sarah, you have another question?
Yeah, I do. Just about dissolution. These are pretty shallow seas, I assume, right? So does that make dissolution less of an issue, or how much do you have to contend with things?
Dissolution is an issue. It's less of an issue in the Pacific than it is in the Atlantic, because the Pacific is just a little bit more acidic, and so the diatoms tend to be preserved, whereas the carbonates are dissolved. But that's not to say that they don't dissolve, because they definitely do. And the dissolution of diatoms, and also the addition of other materials, like, you know, that this one up in the Gulf of Alaska, just filled with stones. And so I actually worked on that one and had to separate the diatoms from the lithic material, which is complicated, and I'm not sure how well it really works. So yeah, dissolution is definitely an issue, which is why IP25 was touted as this great thing, because, you know, it doesn't matter if the diatoms are dissolved. This is always here, except it's only sort of indicating a couple species of diatoms. So it's a little bit problematic, I think.
And
wait, the other thing I wanted to say about that, you said these are probably shallow. This is the weird thing about these sea ice records. almost every single one of these dots, except for a couple up in the Chukchi, are from deep water. And so I think the shallowest site is 800 meters. A lot of these sites are deeper than 1,000 meters, some of them up to 2,000 meters of water depth. So when we think of sea ice today, it's not in deep water. It's only in shallow water. Problem is if you want to get into the shallow water, you have to go up onto the Bering Land Bridge, which is exactly why I was on the Bering Land Bridge last summer, to try to get sea ice records actually from the shelf from shallow water.
Mm-hmm. Yeah, and finally, can you tell the group about the MARDI project and what you guys are doing? You know, just because I think people would be interested. And
so we haven't done very much since we had that meeting. So just Sarah. So basically, MARDI is a PAGES group. So PAGES is the Past Global Changes working group. It's based out of Switzerland, but it's an international group. And so MARDI stands for Marine Arctic Diatoms. And we are a bunch of people from all over the world who work on marine Arctic diatoms coming together. And we've created a database of all of the surface sediments that we have, sort of circumarctic. And then our next step, or what we did, we had one meeting where we talked about the taxonomy. And we came up with the most common species and the most difficult or challenging species. And so what we're working on is starting to put these actually on diatoms.org so that we can have that taxonomy out there. And so we haven't actually done any of that yet. We had this meeting in June, and then all of us just like went out into the field for 6 months. So we're now back, and hopefully some more action will happen. But if any of you are interested in being on our mailing list, I can add you to our mailing list. We're going to have a couple more meetings. One is going to be, one of our plans is to have little micro taxonomy meetings online and talk about some of the different taxa individually. And so that might be something folks here are interested in. And then we'll have another workshop where we talk about like how to bring the data together in statistics and transfer functions and things like that. And as we try to see, like, can we actually take all of this old data and put it together and get something useful out of it that is circumarctic?
Yeah, that's great. Thank you so much.
Beth, I have one more question for you as I think about my participation and work in the Great Lakes. We have a lot of needle-like Fragillaria that end up in the spring plankton and down in the DCL later in the year. And I'm curious how many of, especially just even within Fragillariopsis, as you tease apart the taxonomy more and more, do you have a rough, proportion of how many are obligate and how many are obligate to ice and how many just might be happy accumulating on the ice and living underneath the ice and then maybe in the open water season?
So there are almost none that are ice obligate. There are no Fragillariopsis that are ice obligate. The only ice obligate diatom that I know of that ever shows up in our accounts is Nichia frigida, and that one doesn't show up very often. And so all of these diatoms are diatoms that are happy in the ice, but they can also be just as happy in cold water or, well, cold water.
And before we conclude, it looks like I missed a question from Layla earlier. Layla says, thank you, Beth, for this interesting talk. My question is concerning when you start to have a human effect on the ice distribution from your diatoms. And I'm not sure if she's meaning more globally or from the icebreakers. Layla, if there's any of that you want to clarify, but do you see any sort of direct human influences on the Arctic ice and the diatoms?
So far, no. And the reason for that is because this is too coarse. And so here in the later times, we're only looking at sort of 4,000 year time intervals. And so, people probably showed up in this region Well, we don't really know when they showed up. Maybe they were here all along. There was a paper that came out recently that said that possibly people used sea ice as a sea ice highway to aid in migration from Asia into North America. And so maybe they were out here, but they weren't out here sort of in numbers like, you know, we have today, where we're really impacting sea ice. And my study is not fine enough at this point to be able to say like, oh, this is when we can start to see a decline in Arctic sea ice related to people. I'm guessing it'll probably be right around, you know, the 1900-ish time once we get to that point, just because that's when emissions started, and that would be a huge driver of ice loss.
Well, great. I'll give anybody else one last chance to ask Beth a question as we wrap things up. Thank you very much, Beth. It was great to finally draw you in after 5 years. And I'd like to remind everybody that on January 30th, Rosa Trabajo will be speaking in Espanol, and it will be entertaining, and hopefully we draw in another crowd who maybe is not all focused on speaking English. Let's see, there's a paper in the chat on the ecology of Fragilariopsis and Ficus. Butcher that. I'll open that up and save it. So if anybody else has any last announcements or questions, feel free to chime in. Otherwise, thanks a lot, Beth, for coming out today and talking to us. Appreciate it.
Thank you, guys.
It's great to have you, Beth. Thanks so much. Take care, everyone. See you soon.
Bye-bye.